Welcome back to the Bio 181 lecture series discussing biological macromolecules. In this lecture, we are going to discuss enzymes. We have been focusing primarily on macromolecule groups based on structure or chemical properties, but enzymes are grouped based on function. Enzymes are macromolecules that catalyze or speed up chemical reactions. Although many enzymes are proteins, not all are. Some enzymes, for example, are composed of nucleic acids. Regardless, in this lecture, we are going to talk about common structural and regulatory features of enzymes. Enzymes categorically catalyze chemical reactions. What the heck does that mean? A catalyst is something that speeds up something. I understand this terminology when it applies to enzymes, but I also think that this can be a little bit misleading. Let me explain. In order for a chemical reaction to take place, energy must be applied. The amount of energy that is required for a reaction to take place is called the activation energy. For many reactions, the activation energy required for a reaction to take place is quite high. Thus, it is seen as a barrier to the reaction. In order for the reaction to take place, this barrier must be overcome. Enzymes reduce this barrier, and they reduce the activation energy of a reaction. This would enable the reaction to take place. As you can see on the right, the presence of an enzyme reduces the activation energy of a reaction, thus enabling the reaction to take place. This is why enzymes are referred to as catalysts. They increase the rate of a reaction basically by lowering the amount of energy it would take to enable the reaction to take place. The reason I think that this terminology can be a little misleading is only because the activation energy of many reactions will not be overcome without a catalyst. So yes, enzymes are catalysts and speed up chemical reactions, but in reality, many of these reactions would not take place otherwise. Thus, when we discuss metabolism later, we will discuss several reactions, and we will always discuss the enzymes that catalyze, or as I tend to say, facilitate them. For enzyme function, structure is vital. No way! Enzymes are able to facilitate chemical reactions when a substrate fits into the enzyme active site. A substrate is a molecule on which the reaction will take place. For example, on this slide, the substrate is lactose, a disaccharide composed of the monosaccharides glucose and galactose. In this case, the reaction that needs catalyzing is the breaking of the bond between these two monosaccharides. In order for this reaction to take place, lactose, the substrate, must fit into the active site of the enzyme. It is important to understand that enzymes are absolutely specific for the reactions they catalyze. Furthermore, the active site of an enzyme is absolutely specific for the substrate that fits in it. You can see here that the active site of lactase, the enzyme that catalyzes this reaction, is perfectly suited to accommodate lactose. When it comes to substrate specificity, no other substrate other than lactose would fit in the active site of lactase. Once lactose enters the active site of lactase, lactase positions lactose in a way that lowers the activation energy required to break the bond between glucose and galactose, thus enabling this bond to be broken. So, it is very important that you understand that the active site of an enzyme is where a reaction is catalyzed. The substrate is the molecule undergoing the reaction. For example, Lactose is a substrate that the enzyme lactase will act upon. The active site of an enzyme is absolutely specific for the substrate that fits in its active site. This picture just demonstrates again how substrates fit in the active site of an enzyme. I like this picture though because I think it actually does a better job of accurately representing what an enzyme and its active site might look like in the three-dimensional structure of a protein. I know this is a protein because the individual blue circles represent the individual amino acids. So you can see the complex shape of the protein, which has arranged itself to create an active site for the substrate. When a substrate enters the active site of an enzyme, this is called an enzyme-substrate complex. Sometimes substrates are referred to as reactants. This is because reactants are what enter a chemical reaction. Products exit a chemical reaction. We will talk more about those terms and chemical reactions when we discuss metabolism later. It is very important that in biological systems we are able to regulate the function of enzymes. Basically, we want to be able to make sure that we can control when a reaction takes place. We can do this by essentially giving enzymes on and off switches. Although some enzymes are created in an active state, some are created in an off orientation and thus need to be turned on. There are many ways that this can be done, but one of them is via the process of phosphorylation, basically just attaching a phosphate group to the enzyme. Usually when an enzyme is off, its active site is in a closed orientation, preventing a substrate from entering, and therefore the reaction won't take place. In this case, phosphorylation would just cause the active site to open up, and therefore the substrate could get in and the reaction could progress. Many enzymes, though, are created in an on orientation, and therefore we can regulate them by being able to turn them off. We do this using inhibitors. There are two major classes of inhibitors: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are named competitive inhibitors because they compete with the substrate for the active site. As you can see here, a competitive inhibitor would bind to the active site, effectively blocking the substrate from entering the active site and therefore preventing a reaction from taking place. Non-competitive inhibitors are so named because they do not compete with the substrate for the active site. Instead, they bind to a location called the allosteric site, which is essentially a spot just physically distanced from the active site. Importantly, however, when a non-competitive inhibitor binds to the allosteric site, this does cause a conformational change in the active site. So this still would effectively prevent the substrate from binding and therefore prevent the chemical reaction from taking place. Enzymes are further regulated in a variety of physical and chemical ways. Prosthetic groups, cofactors, and coenzymes are all ways that enzyme function can be enhanced chemically. Prosthetic groups are small molecules that permanently attach to an enzyme. Cofactors are inorganic ions that bind temporarily to an enzyme. And coenzymes are organic molecules that actually participate in a chemical reaction, but are not changed as a result. Regardless of the structure of each of these, they each serve to enhance enzyme function and therefore accelerate the rate of reaction. Other factors can impact enzyme function, for example, homeostatic conditions. As shown in the figure, which is showing the rate of a reaction compared to temperature, increasing temperatures can increase the rate of a reaction to a certain extent. However, once a temperature threshold has been reached, for example, in humans, this would be like above 104 degrees Fahrenheit, enzyme function begins to be impaired. And this is primarily due to the fact that enzymes denature outside of this range. So it's important to note that enzymes function optimally in homeostatic set points. So again, this emphasizes the importance of homeostasis. In humans, as far as temperature regulation goes, this means that your enzymes would function optimally at 98.7 degrees Fahrenheit. Similar effects are seen with pH. Your blood enzymes function optimally at pH of 7, which is the homeostatic set point for your blood pH. The enzymes in your stomach, however, function optimally at around pH 0 to 2, which is the optimal pH of your stomach. Because enzyme structure is so important and macromolecular structure is maintained by homeostasis, the homeostatic set points for an organism or cell are where its enzymes function optimally. When homeostasis is disrupted, so is enzyme function. For this lecture, make sure that you are able to understand that enzymes are catalysts and what they catalyze, understand structures of enzymes and why structure is vital for function, understand how enzymes are regulated, and understand how enhancers and homeostasis and homeostatic defects may impact enzyme function. You do need to know the terms catalyst, activation energy, substrate, active site, allosteric site, activator, inhibitor, competitive inhibitor, and non-competitive inhibitor. Jamie and I are so glad to have you in class, and I will see you again very soon.